Hello everybody and welcome back to Biology 3013. Today's video lecture is going to continue looking through chapter 13, How Populations Evolve. So remember, in part 1 of this video, we looked at the diversity of life and we looked at how there's so many different species. We had to have a way to name these species to eliminate confusion. So that was through the Linnaean naming system. We then took a look at Charles Darwin and his voyage on the HMS Beagle and how he ultimately came up with this theory of natural selection as a way to explain evolution, or how species evolve over time. We finished off by looking at 5 lines of evidence that help to support evolution. Today, we are going to take a little bit of a closer look at natural selection and how it works. We're going to then take a closer look at the genes to see how genes play into how a population is going to evolve over time. And then finish off with a couple of other ways other than natural selection. What are a couple of other ways that these populations can change over time? So let's take a look at section 13.4, where we take a closer look at natural selection. So we've already talked about Darwin and how his trip on the HMS Beagle helped to provide him with the evidence that he needed to come up with this idea of natural selection as a way to explain how species change over time. We talked about some of the influences that played a key role on Darwin and his determination of natural selection. So we mentioned Charles Lyell, who was a geologist, and how Charles Lyell talked about how even Earth's surface gradually changes over time. We saw how there were many naturalists, and Darwin himself saw how fossils looked similar to living organisms, but just with some key differences. And then Darwin also noticed through his observations, through his travels on the HMS Beagle, that he saw that there were many species that lived close together that looked more similar to one another than he would have thought. So we saw that species that lived close together looked similar, whereas species that lived far apart, even if they lived in the same type of environment, they didn't necessarily look the same. So all of those played some influences on Darwin's determination of natural selection. Let's take a look at a couple of other influences. Artificial selection was commonly used in agriculture, and what it is the selective breeding of plants and animals to promote particular traits. So if we think a very simplified example here, and we are trying to have a herd of beef cow that we're going to try to use to have beef. Well, when you are going to fertilize your female cows with these bulls, you're going to try to take that biggest, strongest bull to fertilize your females so that hopefully the next generation of beef cow is going to be larger, stronger, and as a result have more meat on them. So selective breeding has 2 essential components to it. There has to be variation among individuals. So in this simplified example, that would mean that there's lots of different sizes of bulls. There's small bulls, medium bulls, large bulls. There's this natural variation in size, and that size would be heritable, meaning that it's going to be passed down from generation to generation. So again, in our simplified example here, it means that if you take that bigger, stronger bull, then it's going to result in having bigger, stronger offspring. So artificial selection, you could see these fairly large changes in your herd over generations, from these smaller beef cows to these larger, stronger beef cows produced through artificial selection. So Darwin looked at that and he said, well, if you can see such substantial differences in just a couple of generations, well then maybe this same concept can occur in nature, and you can start to see these large changes over thousands or even millions of years. So he started to apply that same principle to evolution. So within a natural population, there is variation. You can see that within this population of ladybugs here. There's different shades of orange, there's different numbers of dots, there's different sizes of dots. So there's this natural variation within the population. So Darwin said, okay, well, the same concept as artificial selection, there is this variation among individuals, but what he didn't understand in a natural environment was what forces in nature were determining which of these ladybugs were going to reproduce and which of them weren't. For artificial selection, you had the farmer that was saying, all right, here's the biggest, strongest bull. This is the one that we're going to take to breed, to use for the next generation. Well, in nature, what was controlling the reproductive success of these organisms? So which one of these ladybugs were going to have the most offspring? So Darwin spent a lot of time thinking about that, and he came upon the work of Thomas Malthus, and he was a philosopher and economist, and he came up with this growth model for the human population that said, over time, the human population is going to grow so quickly, so exponentially, that there's going to be disease and famine and war, because the human population size is going to grow faster than food supplies. So there's not going to be enough food or other resources to feed the entire human population, and as a result, there's going to be famine. So Darwin applied this concept to plants and animals, and what he determined was that he said that, well, if this same concept applies in nature, then there are limited resources. Any environment is going to have a certain amount of food and other resources available for these populations. Well, then the next observation was that if there's overproduction, if there's too many offspring, well, then these offspring are going to struggle for existence. They're going to have to compete with one another for those limited resources. Those that are able to compete and survive, the ones that are able to compete and get the food that they need, those are the ones that are going to survive and reproduce. Those that can't compete, they aren't going to get the food that they need, they're going to end up dying off, and they aren't going to have the chance to reproduce. So this leads to unequal reproductive success, meaning that some of these organisms are going to be very successful at getting the resources that they need in order to have lots of offspring, whereas others of these organisms aren't going to be successful in getting the resources that they need, so they are going to have few, if any, offspring. So a different amount of offspring, an unequal amount of offspring. So that leads us then to our official definition of natural selection. Individuals whose traits are better suited to the environment are the ones that are going to survive and reproduce more successfully. More successfully meaning that they are going to have more offspring, and they're going to pass these traits on to their offspring. So these are adaptive traits that make them that make it so that they are more suited to that environment, and these traits are heritable. They're getting passed down from one generation to the next. So remember, better suited to the environment it means that the physical conditions of that environment are going to control that evolution over time. So individuals better suited to one environment might actually be very poorly suited for a different environment. Let's look at this just very hypothetical example here. Let's just say these are dot bugs, and these dot bugs are living in this white environment here. So if you're looking around this environment, you might say, okay, well, some of these bugs, they are very camouflaged in. So in this hypothetical environment, we have these birds that fly over top, and they like to pick off some of these bugs. Well, the bugs that have this very dark color, they're the ones that are going to be the easiest for the bird to see, so these are the ones that are going to get eaten first. Since they get eaten first, they might not have the chance to reproduce. So if they don't have the chance to reproduce, then that black color that they have there is not going to be very suited to the environment. On the other hand, These white ones, like this one here, is very camouflaged, very hard to see. The birds might not even see that guy at all in order to eat it. So this organism here is going to survive for longer. He's going to be able to mate more often and have more offspring. So over time, because this guy is going to be able to have more offspring, he's going to pass down that white color to the next generation. So if you visualize the next generation, this black guy, he didn't do very well, so there's not going to be as many black colors in the next generation. But this white guy was very successful, so he's going to do very well in the next generation. So there's going to be, there'll be more of these white guys in the next generation. But if we switch things up here and we say, all right, well, now we're in a different environment. In this environment, rather than having this white background, you've got this black background. Well, wait a minute. Now, this white guy who was so successful, this white bug, all of a sudden is the one that sticks out the most. So now this is the one that's going to be the easiest eaten by the bird flying overhead. You're going to easily pick off this white bug. This black one over here, he's very hard to see. So this black one is going to be more reproductively successful. He's going to survive, he's going to reproduce and hand that black color on to the next generation. So now if you picture the next generation in this environment, you're going to have more of these black bugs around and fewer of these white bugs. So it's the opposite as what we saw in the previous example. So what makes a bug reproductively successful or better suited to the environment in one particular environment might not be overly helpful in a different style of environment. So we have to keep in mind that natural selection is always based on the environment that that population is living in. All right, well, let's look at a more realistic example of natural selection in action. So here, this is showing pesticide resistance in insects. So we've got our population of pests here, and we've got our DNA shown here. And in green, we have one allele. In red, we have another allele. This red allele actually makes this bug resistant to pesticides. So when you have a small amount of pesticides, of poison sprayed on these pests, all of these guys with the green allele, they end up dying off. They can't survive. But the ones with the red allele, that pesticide resistance allele, those guys survive, they reproduce, and they're going to hand on that heritable trait of pesticide resistance to the next generation. So generation over generation, those that survive are going to be the ones that are the most resistant to these pesticides. So subsequent applications of these pesticides are going to become less and less effective over time. Okay, so let's look then at a couple of key points that we mentioned that I just want to make sure that it's highlighted. So changes due to natural selection, they're going to accumulate in populations over time. And what I mean by that is that individuals do not evolve. One individual cannot evolve. The smallest level that can evolve is a population. And remember, a population is a group of the same species that are living in the same place at the same time and able to interbreed with one another. So an individual cannot survive because for natural selection to be able to occur, you need to have some of these individuals that are suited to the environment and some of them aren't. The ones that aren't have to die so that those that are most suited to the environment are the ones that are handing down those traits that make it the most suited to the environment. So really death is a key part of natural selection. So one individual cannot evolve. The next key here is that natural selection can only affect heritable traits. It can't affect traits that an organism is gaining through its life. It's only affecting traits that are passed down in its DNA, so that if it has a particular trait and that trait has made it very successful, as long as it's coded for in the genes, you are going to hand those genes down to your offspring, so then your offspring will have that trait to make it successful in the environment as well. The last key here is that evolution is not goal-oriented, meaning that it doesn't have in its mind of a particular end goal that says, oh, this species here, it has to have these particular characteristics and that's going to make it the best suited. It's going to be awesome. Instead, evolution is a result of the environment. So if the environment changes, well then maybe the traits that make it best suited to the environment, those traits are going to change as well. So again, we really saw that with the example of our hypothetical dot bugs, where in the white environment, this white trait was beneficial, but in this black environment, the black trait was more beneficial. So evolution is not goal-oriented. It's not saying, hey, I want all of these bugs to turn black and then I will be happy. Because if the environment changes, the selective pressures change, and as a result, the traits that are most suited to that environment are going to change as well. So evolution is not goal-oriented. All right, so Darwin, one thing that I find fascinating is that Darwin, he saw that there was variation in the environment, and he saw that that some of this variation is going to be passed down from parents to offspring. We know from Biology 3012 that this variation is because of differences within our genes, within our DNA. And we know that these traits are handed down from one generation to the next as we hand that DNA from us down to our offspring. What's fascinating is that Darwin came up with this idea of natural selection, but he didn't know anything about genes or about DNA. We talked about how Gregor Mendel was really important in looking in determining these patterns of inheritance, but Darwin didn't know anything about that. Unfortunately, there wasn't Facebook or Twitter or any kind of social media for these two scientists to be able to communicate with one another. Because if they did, then we would have been able to understand some of these concepts a little bit earlier. All right, so now moving on to section 13.5, where we're going to focus in more on kind of combining Darwin's ideas with what we learned from Gregor Mendel, looking at patterns of inheritance. So remember with Gregor Mendel, we were looking at Punnett squares and how we can look at the traits within your eggs and sperm to determine the chances of having different types of offspring or offspring with different traits. So we're going to combine here a little bit of natural selection with a little bit of patterns of inheritance to see what we get. So let's take a look first at sources of genetic variation. We talked about genetic variation in Biology 3012, and in particular, we were looking at sexual reproduction and how sexual reproduction can lead to lots of variation. So when we were looking at meiosis, we looked at how there are a couple of parts of meiosis that allow for the DNA to get kind of shuffled. So you have information, you have genes from your mom, genes from your dad, and this information can get shuffled through meiosis so that you're going to have a combination of characteristics, or I should use the term traits from your mom, traits from your dad. They're going to get shuffled together within your eggs or your sperm, so your gametes. So remember, this happened through independent orientation of homologous chromosomes. So you can go back to meiosis if that doesn't ring a bell to you. Crossing over, where these homologous pairs can cross over and trade a little bit of information with one another. And then the last step that allows for sexual reproduction to contribute to a lot of genetic variation is through random fertilization. So which particular sperm is going to fertilize the egg and what particular traits is that egg going to be carrying. So through all of these different ways, sexual reproduction leads to huge genetic variation, meaning that if the same two parents have 100 offspring, all of those offspring can be different from one another. They can all have variation in their traits because of the way that sexual reproduction works. Okay, so I won't recap all of the details there, but just know that sexual reproduction leads to genetic variation. All right, well, what else leads to genetic variation is mutations. And we talked a little bit about mutations and how you can plug in the wrong nucleotide in your DNA or your RNA, and that leads to the wrong amino acid being plugged into the protein. Most of the time, those mutations are going to be bad. Most of the time, those mutations are going to lead to a protein that doesn't work, and it's going to end up harming that organism, so the organism is not going to be able to survive. But every once in a while, that new mutation might lead to a new protein that actually helps that organism survive in its environment. Let's go back to our dot bugs to look at another hypothetical example. So if we look here at our population, this guy here, he arose from a mutation. When he was developing, you had the sperm fertilize the egg. This guy's developing. He started off as one cell. He's going through mitosis to become lots of cells. Well, early on in that process, when he was creating new cells, by accident, a mutation occurred, and as that happened, rather than creating the pigment to become gray, instead it produced this new pigment, this new protein that caused it to become yellow. Again, most of the time mutations are bad. So in this environment here, well, now this guy here is going to stick out very easily for that predator, that bird coming through. So it's going to get eaten super quick. So that mutation was harmful, and that new trait, that new color, is going to get eliminated quickly from the environment. But if we have this different environment, if this mutation occurred in this yellow environment here, and now this mutation occurs, and now you have this new pigment, and this organism here is going to now be very camouflaged into its environment. So he's going to be very reproductively successful. He's going to have a lot of time where he's not getting eaten by a bird, so he's going to be able to mate, reproduce, and pass on this mutation, pass on this new trait that's coded for in its DNA because it was a mistake in the DNA. Now it's going to hand that DNA down to the next generation, so his offspring are going to be yellow as well. So then in the next generation, you're going to see more of these yellow dot bugs. And then in the generation after that, you'll see even more of them and even more of them. So slowly, generation over generation, you're going to be able to see that the gene pool changed over time. Now, what I mean by gene pool is the total collection of alleles that are in that population. So remember, alleles are what's in the DNA that's going to correlate to a particular trait. So remember, we have our dominant allele depicted by an uppercase letter and our recessive allele depicted by the lowercase letter. So for this example here, the dominant allele is a red flower color. The recessive allele is the white flower color. The gene pool is saying, how many of these dominant alleles do you have? How many of these recessive alleles do you have? Because the proportion of these different alleles is going to control what alleles are available to contribute to the next generation. So we're going to do a little bit of math in this section of the course. So you will need a calculator for this. You can pause this and grab a calculator now, or you can watch it and then try to use your calculator to do a couple of examples later to make sure that you understand how to do this math. So in the gene pool, P is going to represent the frequency of the dominant allele in the population. Q is going to represent the frequency of the recessive allele in the population. So how many big R's and then how many little r's are there in the population? We know that for any given trait, you've got these two alleles here. If you add the frequency of these two alleles up, then it has to equal the entire population. It has to equal all of the alleles. So P plus Q equals 1 is saying if you add together all the dominant alleles, add together the recessive alleles, that's going to equal all of the alleles, 100% of the alleles. So let's take a look at how we would solve for P and Q. P and Q, when you're looking for the frequency of alleles, you're searching for what's referred to as the allele frequencies. So P and Q are your allele frequencies. So in this example here, I've given you 25 flowers. You can see here they have the different genotypes. Big R is homozygous dominant. Big R, little r is heterozygous, and little r is homozygous recessive. So for these 25 flowers, what are the allele frequencies? So really what I'm saying is how many big R's are there in this population? How many little r's are there in this population? So we know that there's 25 flowers. Each flower has 2 alleles, so we've got a total of 50 alleles. So big R, P represents the big R, the dominant allele. So we're going to just go through and count how many big R's are there in this population. We're going to go through and count how many little r's are there in the population. Once you've counted, you'll see that there's 40 out of the 50 alleles are big R. And if you plug in 40 divided by 50 on your calculator, you'll see that that gives you an allele frequency of 0.8. Q is the frequency of that recessive allele. You can count out how many little r's there are, and you'll see that there's 10 little r's. So 10 out of the 50, 0.2 is the frequency of that recessive allele. Now remember we said that P plus Q equals 1. So if you add together all of the dominant alleles and all of the recessive alleles, that should equal 100% of the alleles. So we add together our 0.8 that we calculated here, our 0.2 that we calculated here, and that gives us 1, and so we are happy. So we have successfully calculated out the allele frequencies for this population. Now, most of the time though, for test questions, we are often given the allele frequencies, and I ask you from those allele frequencies to solve for the genotypic frequencies. The genotypic frequencies are saying what proportion of the population is there for each genotype. So remember, big R is that homozygous dominant genotype. So I'm saying what frequency of the population has the big R? What frequency of the population has big R, little r? And what frequency of the population has this little r? Now, how do we solve for that? Well, one way to do it is by using a Punnett square. I'll go through to show you how the Punnett square works, but you won't actually have to do it as a Punnett square. You will be able to use an equation, but I will show you the Punnett square so you understand where the equation came from. So for the Punnett square, remember, you're going to say, all right, if I have one parent up here, we're putting the mom up here, and the mom has two possible alleles, big R or little r. The dad has two possible alleles, big R or little r. So you're putting those different possible combinations into your Punnett square. Now, last semester, we went through and we said, okay, if you cross a big R with a big R, then we have homozygous dominant. If you cross a big R with a little r, we have heterozygous. Similarly, down here, we're going to have a heterozygous offspring. And lastly, we're going to have a homozygous dominant, homozygous recessive offspring here. Well, to take it one step further, we're going to add in those allele frequencies that we solved for in our last slide. So the allele frequencies, we're saying, well, how often do we have big R in that population? Well, 0.8 is the allele frequency there. How often is it little r? 0.2. So then when you multiply these numbers together, 0.8 times 0.8, you have 0.64. So what that's saying is that 0.64 or 64% of the time, you're going to have this homozygous dominant genotype. Well then here, if you multiply out 0.2 times 0.8, you're going to get 0.16. Down here again, you're going to get 0.16. So if you add those together, 0.32, shown down here, 0.32 or 32% of the time, you're going to have big R, little r, that heterozygous genotype. Lastly, down here, multiply out your 0.2 times your 0.2, 0.4 or 4% of the time, you're going to have this homozygous recessive genotype. And if you actually take those numbers and you apply it back to our flower population from the previous slide, you will see that those numbers are accurate, where 64% of those flowers, 64% of them, or 64% of 25 flowers would be 16. And if you count it out, 16 of these flowers are big R, big R. 32% would be 8 of these flowers are heterozygous. And if you count them out, there are 8 heterozygous flowers here. And lastly, 4% would be homozygous recessive or little r, and that would be 4% would be 1 flower, and there is 1 flower here. So this population here, the gene pool from this population controls the next generation. Now, here we solved the genotypes, or the genotypic frequencies, by using a Punnett square. But now that we've done this Punnett square once, we can actually just use these values here. So it gives us the equation p squared plus 2pq plus q squared equals 1. So the p squared was taken from here, the 2pq were taken from these two boxes, and the q squared taken from this box here. So the p squared in the equation represents the frequency of the homozygous dominant genotype. The 2pq represents the frequency of the heterozygous genotype, and the q squared represents the frequency of the homozygous recessive genotype. So now, if you are given just the allele frequencies, just from the allele frequencies, you can use this equation to solve for the genotypic frequencies. Let's complete an example of that. So with this example here, we're saying we've got this PKU, fetal ketonuria, which is an autosomal recessive disease, meaning that the disorder is carried on the recessive allele. And this occurs in about 1 in every 10,000 babies born in the US. 1 in every 10,000 babies have this disease. So from that information, it's saying calculate the frequencies of the different genotypes. And in particular, public health scientists often use this equation to figure out the proportion of the population that are going to be carriers for a particular disease. So based on what is given here, let's try to write out what we know. So we know that 1 out of every 10,000 babies born have PKU, so they have the disease. We know that this disease is caused by the recessive allele, so it's carried by the recessive allele, so that's the lowercase letter. Now I chose the letter D here just for disease, so the uppercase D would be for the healthy allele and the lowercase d would be for the diseased allele. You can choose any letter that you want, but it's just remember the uppercase is the dominant and the lowercase is the recessive. So since it's carried on the recessive allele here, we know that it's carried by that little d. All right, well then in general, we know that there's 3 different possible genotypes. So if it's saying calculate the genotypic frequencies, well, we want to get the frequencies for these 3 genotypes: homozygous dominant, heterozygous, and homozygous recessive. Well now, when we think of these genotypes, we know that genotypes control the phenotypes. So what phenotypes do these organisms have? Well, the homozygous dominant, that big D, is that healthy allele, so those organisms are going to be completely healthy. The heterozygous, they have 1 healthy allele and 1 diseased allele. So these guys are going to be healthy, but they're going to be carriers. They're going to be the organisms in the population that are going to be able to pass down this allele to their offspring. The last genotype here is our homozygous recessive. Those are the organisms that are going to have the disease, that are going to have PKU. Okay, so then, with this information, we know that 1 out of every 10,000 babies have PKU, and now we know that it's that homozygous recessive genotype that means that the organism has PKU. So that must mean that our homozygous dominant, or our homozygous recessive genotype is what's going to equal 1 out of every 10,000. Now, when we look at our Hardy-Weinberg equation, we know that the p squared represents the frequency of the homozygous dominant genotype. Our 2pq represents the frequency of the heterozygous genotype. The q squared represents the frequency of the homozygous recessive genotype. So that's our PKU. So q squared is what's going to equal our PKU, or q squared is what's going to equal our 1 out of every 10,000 babies. So now we can start to solve for this question. So we know that q squared is going to equal 1 out of 10,000. You can plug that into your calculator. 1 divided by 10,000 equals 0.001. Well, if q squared equals 0.01, we want to find out this number here, our frequency of heterozygotes. We want to figure out this number here, our frequency of homozygous dominance. Well, in order to do that, we need to be able to solve for P and solve for Q. Right now, all we have is Q squared. Well, if we know Q squared, we can solve for Q. So Q squared equals 0.0001. Q, if you do the square root of that answer, you can solve for Q. So Q will be the square root of 0.0001, which gives you 0.01. So Q equals 0.01. Now don't forget what that represents. Q is our allele frequency. So P plus Q, our homozygous dominant allele here, or pardon me, our dominant allele is represented by P. Our recessive allele is represented by Q. So 0.01 of all of the alleles in the population are that recessive allele. P plus Q equals 1, so if you rearrange that formula, then P equals 1 minus Q. So 1 minus 0.01 is going to give you 0.99. So that's going to give you your other allele frequency. So you're going to have 0.99 of all of your alleles are going to be that dominant allele. 0.01 of all of the alleles are going to be that recessive allele. Now that we have those P's and Q's, those allele frequencies, now we can solve for the genotypic frequencies. So in order to do that, we're going to use the different components of our Hardy-Weinberg equation. So we know that P squared represents the frequency of the homozygous dominant genotype. So we're going to plug in P squared. Our P was 0.99, so 0.99 squared, or 0.99 times 0.99 is equal to 0.9801. Again, you can just plug that into your calculator. The frequency of heterozygous genotype is depicted by 2pq. Plug in the numbers that we solved for in our last, in our allele frequencies on our last slide. 2 times 0.01 times 0.99. Plug that into your calculator and get your answer. Now remember, the heterozygotes, those are the ones that are going to be the carriers in the population. So if a question asks you particularly to solve for the frequency of carriers in the population, then you're just focusing in on this one aspect of the Hardy-Weinberg equation, solving for that 2pq. Now our third genotype, the homozygous recessive frequency here, we already knew that one. That's the one that was given to us out of 1 out of every 10,000. So our, this should have 2q squared, sorry. Let's plug a 2 in here. So q squared is going to equal 0.001. That's the 1 divided by 10,000. That's what was given to us in the question. So we don't really have to solve for that, but it's good to write it out so that we have all 3 of our genotypic frequencies here. Now the last step that you should always do is to just double check that you did it correctly. So take the frequencies that you solved for, take your P squared, take your 2pq, take your Q squared, and add them together. If you've done it properly, then it should equal 1, and we will all be happy. But that being said, sometimes you will have rounding errors. We don't talk about significant figures in this course, so I can't tell you exactly how many decimal points, but I would recommend using at least 4 or so decimal points within these questions. And then if you get very close to 1, if you're kind of 0.999 or 1.001, then you're going to be pretty close, so you know that you probably did it right and just had just rounded off a couple of those digits so that it was slightly off, but that's not a problem. As long as it's close to 1, I'll still give you the value for that question. All right, so you've solved for the allele frequencies, you've solved for the genotypic frequencies, you've double checked your work, and I am happy, and you get 100, so you are happy too. Now to put this back into perspective, why are we doing all of this and how does it relate to evolution? I thought we were talking about evolution here. Well, we said that the allele frequencies are making up the gene pool. And it's the gene pool that's going to be the raw material for the next generation. So you can look at changes in this gene pool to see if that population is evolving. So you do that Hardy-Weinberg equation and you figure out those genotypic frequencies. And if those frequencies aren't changing from one generation to the next, then you're in what's called Hardy-Weinberg equilibrium. That gene pool is remaining constant, so it's not evolving over time. If you do find that generation after generation you're solving for these genotypic frequencies and you're seeing changes in this frequencies over time, well then that's evolution. And in particular, microevolution is where you have these small scale, these generation to generation changes that you can see within the frequencies of the alleles and as a result, the frequencies of the genotypes as well. So you can use this math, this Hardy-Weinberg equation, in order to determine whether a population is changing over time. All right, so that was a mouthful. There's a lot of information there. Some students find these questions super easy. Some people find them quite difficult. So do a couple of practice questions. And if you have any questions about it, then send me an email and I'd be happy to review it with you to make sure that you fully understand it. Now we're going to move on to section 13.6. In this section, we are looking at different mechanisms of evolution. So we've already talked about how evolution is these populations changing over time, and we already talked about Darwin saying that natural selection is the mechanism for evolution, but there are actually a couple of other ways that these populations can change over time. Genetic drift and gene flow are two other ways that these populations change over time. I do want to point out though that natural selection is the most important one because it's the only one that's promoting adaptation. It's the only one that's making the population more suited to its environment. But let's take a look at the other two and what they are. So genetic drift is when the population changes over time. So you have a change in the gene pool, and that this change is because of chance. So it's not because certain organisms were best suited to the environment. The organisms that are in the population are only there because of chance events. A good analogy to use for genetic drift is a coin flip. We know that a coin has two sides, heads or tails. So if you flip that coin a large amount of times, like let's say 100 times, you should have roughly 50 heads and 50 tails. But if you reduce that number of times that you're flipping, reduce that sample size just to 10, well then there's going to be a higher level of chance involved. So you might get 5 heads and 5 tails, or by chance, on that time that you're doing it, maybe you get 7 heads and 3 tails, or maybe you get 1 head and 9 tails. The smaller sample size, you're not going to, or you may not have that predicted outcome. So with genetic drift, if you reduce that sample size, reduce that population size, well then you might not have the same allele frequencies. So you could have a random change in the gene pool when you reduce that population size. So how does that population size shrink? Well, there's a couple of ways that this happens. One is the bottleneck effect and the other is the founder effect. The bottleneck effect is when you have a drastic reduction in your population size that's normally resulting from some sort of natural disaster. So something like an earthquake or a flood or a fire. And the results are that the population that's left over is going to have a different gene pool. So you're not going to have those same allele frequencies as the original population. You might even lose some of that genetic variation. So some alleles that you had in that parent population, those alleles might have gotten wiped out so that those alleles aren't remaining within the gene pool. Let's look again at our hypothetical dot bug population here. So in this population, we had a new allele that arose because of a mutation, and that allowed for this yellow trait. Well, let's say that fire came through and killed off a lot of these bugs. Well, if you killed off this bug here, well then you're losing some genetic variation. You're losing that allele in the population. So that's going to affect the gene pool, and it's going to result, again, it's a result of random changes or changes due to chance. An analogy to this is if you take marbles in a bottle, and these marbles represent the original population, the different numbers or the different frequencies of purple and green and orange are going to represent your different allele frequencies. You dump a few of these marbles out, this is your surviving population after the bottleneck event, and you can see here your gene pool is very different. Your allele frequencies are going to be very different, and you've even lost some of that variation because you've lost that orange allele. So that's the bottleneck effect, where you're going to have often natural disasters, but there are other examples. For example, human hunting, where we go through and wipe out the majority of a population. These are different ways that you're going to change the gene pool of that population. The founder effect is another way, another type of genetic drift. And the founder effect is when you're going to have a few individuals from a parent population go and colonize and start a new population. So then the gene pool in that new population is going to be different from the parent population. And again, because you have just a few individuals going to colonize that new population, this again, the gene pool that you have as a result is just going to be associated with chance. As an example here, there were 15 people who colonized Tristan da Cunha, and one of those colonists was carrying a recessive allele for this progressive form of blindness called retinitis pigmentosa. And this one person was just a carrier. So they didn't have this blindness. Nobody was aware of it, but that person carried the allele for this blindness. And then 150 years later, the 240 descendants on the island, 4 of them had this progressive form of blindness. 9 of them were carriers, and these numbers were 10 times higher than the original population. So you had a change in the gene pool of this new population as compared to the original population, and this was due to chance. Our second way that populations change over time is due to gene flow. And gene flow is when you have genetic exchange with another population. So maybe you have somebody coming into the population that has different alleles, or maybe you have somebody that's leaving the population. As the population gains or loses alleles, that in itself is going to change the gene pool. Well, then on top of that, rather than entire organisms coming and going, you can also have gametes that are going to flow from one population to another. So if you think of plant pollen, the plant pollen or the sperm is going to blow to a new population, and that's going to introduce different alleles or changes in allele frequencies to the other population that it blows into. So you're going to have a different gene pool. Again, this isn't going to make that population more suited to the environment, but the end result is really going to be a reduction in the difference between these populations over time. Anytime I think of gene flow, I always think of Time magazine, who a couple decades ago now, in 1993, they came up with a cover that was an image of a woman who is computer generated from a mix of all kinds of different races to show the new face of America. And it was talking about how as humans were more freely moving around the world from continent to continent, these continents, they were more interconnected than ever. And with people that were moving from one continent to the next, it meant that the gene flow, or the gene pool between these populations, they were all changing. And the genetic differences between the populations on these continents that used to be more isolated, well, these genetic differences were starting to get smaller and smaller over time. So that's gene flow. The third way that populations are going to change over time is the one that we've covered through most of this chapter, is natural selection. And again, this is the only one that's resulting in a population that's better suited to its environment. So an example of natural selection, we've gone through a couple, but you can see in this example here with this type of bird called the blue-footed booby, you can see how it's become quite adapted to its environment. It's from the Galapagos Islands, and it has a body that's really streamlined. It's surrounded by water, so it's got a body that's streamlined to be able to dive into the water, to be able to search for food. It has this large tail that can actually function as a brake, and then the males actually have these bright blue feet because the female birds prefer mating with males that have these bright blue feet. So that's a type of sexual selection. We'll take a closer look at that in a couple of slides as well. So natural selection is going to change the traits of this population and the frequencies of the traits of this population over time. Now, really, there's three different ways or three different outcomes of natural selection. So this is saying how natural selection is going to affect the phenotypes of this population. Directional selection, disruptive selection, and stabilizing selection. And we're actually going to take a look at this graph here to explain those terms. So here, the original population, this bell curve is showing that you have a few organisms that have this extremely white phenotype, or this is showing color of mice, but it can be any phenotype that you're thinking of. So it could be body size, it could be body color. The most extreme phenotype is going to have just a few organisms that have that particular phenotype. You're going to have the majority of the organisms, a higher frequency of organisms that have that intermediate phenotype, and then you're going to again have a smaller amount of organisms that have the opposite extreme phenotype. So if this is body size, then you're going to have a few that are really small, most of them that are kind of medium, and some of them that are really big. So we know that this comes down to that natural variation that we talked about that occurs within natural environments. You're going to have a natural variation in the organisms within that population. So natural selection is going to shift this curve in some way. Directional selection is saying that you're going to push that curve in one direction. So you're in this example here, the dotted lines are the original population, and over time, the population evolved to become darker. With this example, you can think of the environment changing. So I like to think back when I was in university, we looked at how moth colors can change over time. And in particular, they were closing down a coal processing plant. And so we were looking at moth colors before the coal processing plant closed and then after the plant closed. And what we found was that because the coal processing plant kind of created this black soot that would land on the trees, the moths had this darker color. The darker color allowed them to camouflage in, so the darker color was best suited to the environment. After this processing plant closed, well then there was no longer this black soot being emitted. And as a result, you saw this gradual shift in the phenotypes over time where you were having a lighter phenotype that were favored. So you had this directional selection. In the example of the moths, the directional selection occurred in this way here, where it was favoring a lighter phenotype. But it can happen in either direction. You're going to favor one of the extreme phenotypes. Disruptive selection is when you're going to have natural selection that's going to favor the two extreme phenotypes, and the intermediate phenotype is actually not advantageous. So this is common in something like in a patchy environment. So if you think of, let's just go with the hypothetical environment where you have really white sand, and then you have these black rocks studded amongst that sand. And these mice, they can either camouflage into that white sand, or they can camouflage in next to those black rocks. So it's advantageous to be white or black, but it's not advantageous to be the color in between. If it's gray, then you can see it on the sand. You can also see it by the rocks. So it's not advantageous to have that intermediate phenotype. Stabilizing selection is the opposite of that. Stabilizing selection is the one that's actually by far the most common. And it's the one that's saying that that intermediate phenotype is the one that's the most favored for. So you're not going to have nearly as many of either of the extreme phenotypes, and the majority of the population is going to have that intermediate phenotype. Stabilizing selection is actually what keeps human births. Most offspring, most children that are born, are typically between 6.5 to 9 pounds. And that's because anything below 6.5, you're going to have a higher mortality rate. Anything above 9, you're going to have a higher mortality rate. There's going to be some there, but the stabilizing selection is going to favor those babies that are within that intermediate birth weight. The intermediate birth weight is going to be the one that has the highest chances of survival. Now, for humans, because we have medicine and hospitals, we can kind of intervene on that a little bit to be able to help those with a low birth weight or a high birth rate to be able to survive. But if you think of more natural populations that are giving birth outside of hospitals, something like these mice or any population, then you can see how the same concept would apply, where the mice that are born that are really too small, they're going to have a hard chance of surviving. Those that are really too big are going to have a hard chance of surviving. Maybe it's really hard for the mom to give birth to those because they're so large. Maybe they can't get enough food because they're so large. So stabilizing selection is saying that those offspring that have the intermediate birth weight are the ones that are the most likely to survive. Okay, so these are the 3 different outcomes of natural selection. The last slide here just has a quick note on sexual selection. Sexual selection is why we said that those blue-footed boobies have their blue feet. And it's a form of natural selection where individuals with certain traits are more likely to reproduce because of typically the females are going to select which male to mate with. There are some examples where the male is selecting which female to mate with, but most often in the animal kingdom, you have the females that are going to choose which male to mate with. And so you end up having sexual dimorphism, which means that di is standing for 2, morphism or morphology is appearance. So you're going to have 2 appearances depending on the gender, the sex of that organism. So males and females are going to look different from one another. And the reason they look different is because of the different selective pressures, meaning that the females are going to be able to choose which males to mate with, and the females might say, hey, I want to mate with the male that has the brightest colored plumage. So the males over time, the males that have the brightest feathers, they're the ones that are going to survive and reproduce. For females, there's no advantage to them having those bright feathers, so they won't have those bright feathers. So you end up having males and females that look different from one another. So sexual selection just ties into natural selection and how these populations can change over time. All right, that finishes off chapter 13. As always, if there's anything that I covered that you still don't understand, please reach out to me by email and I will be happy to help you out. So other than that, good luck with your studies.